I don't wanna grow up, I'm a curious kid. The world has 100 questions I can play with. So I'll open my arms and eyes and wonder every day till the day I die. No one really knows why gravity exists.
Good evening. My name is Evan Groover, and I'm a PhD candidate at UC Berkeley in the Department of Plant and Microbial Biology. Thank you so much for coming out to my talk titled CRISPR Crops, Food, Farms, and the Shape of Plants to Come. Tonight, I'm going to talk about some big ideas in plant science, which is my field, also in agriculture, and in gene editing, specifically with regards to a new tool called CRISPR-Cas9 that is allowing biologists and crop breeders alike to really progress the way that we're able to generate new diversity in crop plants. Now, really before I get started, I wanted to take an opportunity to thank Wonderfest, who has been so awesome in facilitating this whole event. If you don't know, this was supposed to be a live event, and they have been really amazing in facilitating the transition to a YouTube event. So thank you to them, and thank you to you for your attention as we get going. So a lot of the ideas that I'll talk about today have to do with this graph. And as a plant scientist, I find myself thinking about this graph pretty frequently. What this shows is a UN estimate of our human population in just 30 years' time, in 2050. And what we can see is that our human population is set to exceed 9.7 billion people. Feeding that many mouths will mean that we'll have to practically double our agricultural output from where it currently sits. And in doing so, we're going to have to do it on even less land than what we're used to, as to mitigate deforestation. From almost every estimate, meeting this challenge will probably be the most significant hurdle faced by humanity in the next 30 years. But today, I'm here to tell you that there's hope. I'm going to tell you about a revolution in agricultural productivity enabled by gene editing and genetic modification, and the stories of the farmers and scientists fighting on the front line against climate change. Now, you might have heard that gene editing or genetic modification of agriculture is some nefarious force, and you're not alone. A lot of Americans think that. But today, I'm here to give you a different perspective. I'm here to tell you that these tools are going to be absolutely invaluable in improving our food for human safety, for meeting the needs of a growing population while protecting the people that grow our food, and also for really preserving and sustaining our environment in the way that is most ethical. I'm going to tell you the story of where our crops have come from and how domestication has taken place in crops historically. And then I'm going to tell you about a new tool called CRISPR-Cas9 that's revolutionizing the way that farmers and scientists are able to grow crops for sustainability, for nutrition, and for safety. I'm going to tell you how CRISPR and genetic modification is changing the food that we eat, the way that we farm, and the way that we could be using plants to meet the needs of a changing climate. And finally, I really hope to impart in you how your opinions and your perspectives can be absolutely instrumental in enabling this societal revolution kind of brought on by CRISPR-Cas9. And I hope to kind of inform your perspective on that a little bit with the evidence that I'll present today. So to talk about where we're going in agricultural improvement, I think it's very important to talk about where we've been in agriculture. And that's why I like to start with still lifes. So I ask that you consider this still life from the 17th century in Italy. What we see is a lot of fruits, stone fruit, pears, apples, they all look pretty similar to the things that we're used to seeing today. But this watermelon looks entirely wacky compared to what we're used to. It's mostly rind, it's full of seeds, and it looks hardly edible compared to the things that we're used to eating. Fast forward 150 years, and things haven't really improved for the watermelon. This was a still life that was actually done in the US, so this is a whole continent away, and humans still really haven't gotten the whole watermelon thing down. Now, what this really tells us is that much of the improvement that has made watermelon seedless and fleshy and delicious has really occurred in the past 100 years. And what I mean to highlight here is that agricultural improvement, the actual, modification of our crops to be more suitable for human consumption, is a very active process. Now, while this process has been going on for thousands of years, there's still a lot of huge improvements to be made. And today I'm going to tell you about that process of domestication, how we've got here, and then also some of those improvements that we can make with new genetic modification technologies. Now, you have to imagine that if you were a human about 13,000 years ago, there really wasn't even agriculture yet. Humans were mostly nomadic at that point, and while they had traveled to most of the continents of the Earth, they were still kind of roaming around, picking berries, hunting animals, being, traditional hunter-gatherer types. Now, this all changed in about 11,000 BC in what was called the Neolithic Revolution. And this is when a group of humans in modern-day Turkey, Iraq, and Syria, what we call the Fertile Crescent, descended upon an area that they saw was very suitable for domestication of crops. And so they found wild species of grasses, like einkorn wheat, which we see here, and actually started breeding them together to make them more suitable for producing food for humans. This wasn't just limited to crops. Humans actually also figured out that they could domesticate large animals, and they also started, setting up complex systems of irrigation where they were growing fishes, essentially established all of these primordial technologies that really brought human society together. And really it was agriculture and domestication of wild species that were actually the foundation of human civilization. What's interesting about this phenomenon is that it wasn't really unique to the Fertile Crescent. And if we look all across the Earth, we can see in the following centuries that humans actually independently discovered agricultural domestication multiple times and in multiple regions of the Earth. And so just as soon as wheat and barley had been domesticated in the Fertile Crescent, we can see in Africa things like sorghum and peas and types of beans were being domesticated. Simultaneously, rice was being domesticated in East Asia. If we look all around the Earth, we can see different signatures of agricultural crop domestication, where wild crops were turned into foodstuffs for humans that could be grown successively over multiple years. Now, across human civilizations, some of the first wild plants to be domesticated were cereal grains. So this is where grasses were turned into things like wheat and barley and maize and sorghum. And so a really extreme example of this is teosinte, which is this tiny little grass that grows in parts of Mexico that over several millennia was domesticated into modern corn, which we can see here. Besides the domestication of grasses, the domestication of fruits was also really central to the foundations of society. Here we see a really good example in banana, which, if you look at the wild variety, it looks practically inedible for the amount of seeds that it has and how thick and kind of, rough its skin is. And over many generations, mostly in Papua New Guinea and then later in other parts of the world, banana was domesticated into the delicious fruits that we have today. And so what I hope this imparts in you is that agriculture has really never been a natural process. For as long as we've been growing foods as a part of our society, agriculture has been an intentional process of modification of plants. Now, in the millennia following the initial domestication of crops, humans were able to make really great strides in the food that we eat and the way it was able to be farmed, and this was all happening through conventional breeding, where one plant was crossed by another and their offspring would take on some of the traits of its parents. But up until the 1800s, no one really understood the true mechanics of that inheritance. This all really changed in the 1850s with Charles Darwin's theory of evolution. Darwin was able to show that there actually existed really sound evidence for the transmission of traits in a predictable manner from parent to offspring, but also that this phenomenon, this evolutionary phenomenon, really accounted for most of the things that had been observed in the domestication of crops and in the evolution of wild organisms. This theory was really greatly expanded on by an Augustinian friar named Gregor Mendel, who was able to show the exact mechanics of how traits were passed from parent to offspring. He did this by breeding peas together and seeing how traits segregated from generation to generation. Now, what Mendel had really discovered was this concept of a gene, of a fundamental unit of inheritance that passes between generations. While he had described genes very eloquently, no one really understood what they were made of, and in the decades following his work, scientists sought to understand the material basis for the gene. This all came with the discovery and the characterization of DNA. Now, newfound understanding of DNA and genetic inheritance had huge implications on the way that humans were able to improve crops. And perhaps the most notable historical example of this is a period called the Green Revolution from 1950 to 1970. The Green Revolution was essentially an international technology transfer initiative where Americans, mostly American philanthropic organizations and agronomists, passed a bunch of expertise to the developing world to help them increase their agricultural output. Now, beyond the use of kind of agrochemical techniques, so that's like fertilizers and pesticides, the Green Revolution mostly dealt with breeding crops for improved pest tolerance, drought tolerance, and yield. And so if we look on the right here, we can see this kind of stunted dwarf variety is actually a really improved variety of wheat that was championed in the Green Revolution. The implementation of dwarf varieties of wheat and rice actually is said to have saved about 1 billion people in the period from 1950 to 1970. If we look at just wheat yield alone in some of the Green Revolution countries, we can see that the implementation of advanced breeding techniques really improved output. And this actually wasn't just in the developing world. And so I tell you about the Green Revolution for essentially 2 reasons. And the first is that it's a great example of how when technology improves, we can really do great things for agriculture. And the second reason I tell you is because it's an excellent example of how American philanthropic organizations can really spearhead agricultural improvement in the developing world, which I will make a case for as being absolutely essential in the coming decades. Beyond enabling a huge transformation in agronomic productivity in the Green Revolution, the understanding of DNA inheritance also had huge implications on tools that we could use to improve crops. One kind of quirky example of this is called atomic gardening. So after World War II, American scientists and politicians were looking for new ways that they could use atomic energy for peacetime solutions. And one of the things that they thought to do was to set up these huge gardens full of thousands of plants that they would then irradiate with gamma radiation. And what they found is that when you systematically mutagenize a lot of plants, sometimes you'll produce new varieties that have traits that are appealing for farmers or consumers. While atomic gardening is really quirky, you might be surprised to learn that over 1,000 varieties that are currently sold in stores have been generated with mutagenesis-based techniques like this. So that means if they weren't just using radiation, maybe they were using chemical mutagens or other tools for systematically and randomly mutagenizing the genomes of plants. Beyond random mutagenesis, humans also started to discover new molecular biological techniques for the insertion of genes into genomes of organisms. This mutagenesis breeding technique, like atomic gardening, and then also the insertion of foreign genes into plants, became pretty popular in the 2000s as new means of producing diversity and kind of desired traits into crop plants. But still, the great feat of genome engineering, of gene editing, remained elusive, and that is to be able to very intentionally and specifically modify a gene within a very, very precise point within an organism's DNA code. This all changed with the discovery of CRISPR-Cas9. CRISPR-Cas9 is a molecular biological tool that lets scientists and plant breeders specifically modify the genomes of living things at a designated site inside of their long DNA code. CRISPR-Cas9 was discovered in about 2011, right here in Berkeley, and has seen incredible popularity as a new tool for biological exploration and crop improvement. But it actually wasn't really discovered for the sake of being a genome editing tool. It was discovered as a constituent part of a bacterial immune system. And so, in perhaps what is something that is very relevant to our whole COVID thing going on right now, it's worth pointing out that besides humans, bacteria also are in a constant war with viruses that exist in their environment. And for bacteria, these viruses are called bacteriophage. And just like us, bacteria have an adaptive immune system that is able to be exposed to a virus, then learn to recognize that virus and kill it upon subsequent exposures. For bacteria, that immune system is called CRISPR. So let's take ourselves inside of a bacterial cell and learn what happens when it's infected by a bacteriophage. Usually a bacteriophage will land on the surface of that bacterial cell and insert a bit of DNA inside of the bacterium. Now, in most situations, this is the end of the story. The virus replicates itself inside of the bacteria, causes the bacteria to explode, and all of those progeny viruses go off into the population and take out more bacteria. But in certain situations, when a CRISPR immune system is present, the bacterium is able to recognize that viral DNA, grab it, and embed it inside of its own genome, inside of its own DNA code, between a series of very kind of repeated and palindromic pieces of DNA. These palindromic pieces of DNA are called clustered, regularly interspaced, short palindromic repeats, or CRISPR. Now, CRISPR, these actually repeated sequences of DNA, had been really speculated about and observed in the microbiological literature for decades. But it wasn't until about 2012, right here at UC Berkeley, when a team led by Jennifer Doudna and Emmanuel Charpentier discovered that they were involved in host immunity against viral infection. They found that when that viral DNA is taken up into the genome of the bacterium, it's used to entrain a series of protein molecules that float around the cell, surveilling for subsequent exposure to a viral DNA piece. Once that viral DNA is put inside of the cell, those protein surveillance molecules grab onto the DNA and destroy it, effectively chopping it into bits. It was our team of scientists at UC Berkeley that was able to see the potential of that surveillance molecule, a programmable DNA cutting enzyme that they named Cas9, or CRISPR-associated protein 9. Now, Cas9 is a programmable, sequence-specific DNA cutting enzyme, which means that it can find a specific point of DNA and cut it. So consider a leaf cell. If you drill way into a leaf cell, you'll get down to its DNA code. What Cas9 is able to do is sort of scan along that genome and find a specific point, usually 18 to 22 letters long, sort of like a Control F function, and then cut it in half, sort of like a Control-X function. Shown a little bit more pictographically, we can see that Castline essentially scans along the genome until it finds that very specific sequence to which it was entrained. Once it finds that sequence, it's able to pull the DNA apart, cause a cut, and essentially make a cleavage in both sides of that DNA. So when you cut the genomes of living organisms, lots of different things can happen. But let's consider those two things that are most specific to agricultural improvement. The first thing that you can do is use Castline to create a mutation. And so this is where we cut the DNA and delete, alter, or change the activity of a gene inside of that genome. And under the current regulatory paradigm, this is considered gene editing. It's not really regulated as genetic modification. The rationale behind this is that if I wanted to, say, create a mutation in a specific gene, I could A, take 1,000 plants and irradiate them with gamma radiation or something similar to that, or I can just take one plant and hit it with Cas9 and do it a lot faster and less expensively. And so, essentially, Cas9 is just a more efficient way of producing site-specific mutations, and that's one of the ways that we can use it. The second way that we can use it is in a process called gene insertion. And so that's where we supply a piece of donor DNA along with our Cas9 that is able to actually fill that gap that's created by a DNA breakage event. Now, in most cases, this is used to insert a gene inside of the genome of living plants, or it's used to kind of alter the activity of an existing gene. But because we're supplying foreign DNA that's inserting itself into the genome, this is considered true genetic modification and is regulated as such. When you use this technique, you produce a GMO, or genetically modified organism. Now, today, I hope that I can convince you that both gene editing and genetic modification are safe, practical, and much more efficient than other breeding techniques, but also that they'll be absolutely critical in meeting the needs of a growing population and changing climate. But first, let's start with the food. Now, we all eat food, and I think a lot of people have very warranted skepticism about how genetic modification could be used because they're concerned about keeping their food nutritious and safe. And it's my job as a plant scientist to tell you that many of the tools that we're using for genetic modification and the techniques that we're applying are to really improve the nutrition and the safety of our food for human consumption. I'm going to give you a few examples of that right now. First, let's consider tomatoes. Now, you, like me, might see this image of grocery store tomatoes and kind of reel back in your seat. And it's not because I don't love tomatoes. In fact, an heirloom tomato picked in the middle of summer is my absolute favorite fruit ever. But it's not really an unpopular opinion that most of the tomatoes that are sold in the grocery store are cardboardy, flavorless, and lack a ton of nutrition compared to heirloom or wild tomato varieties. The main reason for this is because over the past 100 years, tomatoes have been mostly bred to be harvested in industrial agricultural environments, such that they've lost a lot of their flavor and a lot of their nutrition. But what if we could use Cas9 and advanced breeding techniques to kind of redo that domestication process? What if we could start with a flavorful and nutritious wild tomato and use Cas9 to domesticate it to make it more attractive for farmers? 2 years ago, one team showed that we could do just that. They went back to basics and started with an undomesticated variety of wild tomato called Solanum pimpinellifolium. Solanum pimpinellifolium is delicious, but its fruits are very small and they grow on a huge bush, which would never be attractive for farmers because it couldn't really fit in a field. So, they decided to use Cas9 to mutagenize those genes that were most associated with domestication to make those tomato plants attractive but still delicious and nutritious. Using Cas9 and mutagenizing just 4 genes, our scientists were able to replicate a domestication process that took farmers thousands of years using conventional breeding. By mutagenizing one gene, they improved the architecture of that wild tomato plant, turning it from a big bush to a much more compact one that would be attractive to farmers. With a second mutation, they were able to increase the fruit number on that plant almost 10 times. With a third, they were able to increase the size of those fruits, such that they looked much more like the ones that you could buy in the store today. And with the fourth mutation, they were able to improve the expression of lycopene, a critical nutritious chemical that's found in most tomatoes, but has been lost in a lot of cultivated varieties that are sold in the store today. At the end of all of this gene editing, our scientists had produced a variety of tomato that looked very similar to the ones that are sold in the store today, but it had retained all of the flavor and nutrition of a wild tomato variety. I think we can all agree that this might be a really exciting application of gene editing, but it could have implications far beyond crops like tomato. Consider orphan crops like cowpea, yam, millet, and cassava. Crops like these are absolutely central to meeting the nutritional calories of billions of humans worldwide, but by most standards, they're relatively undomesticated, which means that they have many unfavorable traits that makes them hard to grow and sometimes unattractive for consumers. I ask that you consider just cassava, a starchy root vegetable that's central to the diet of 40% of Africans and is a staple for about 1.6 billion humans worldwide. Now, as an orphan crop, that means that cassava really hasn't received a lot of the same attention from crop breeders and is by some standards undomesticated. But cassava especially has a huge problem. That problem is that cassava plants produce a cyanide-containing compound inside of their roots, which is like the true edible portion of cassava plants. Cassava plants are also very hardy, so that means that in many parts of rural Africa, when drought occurs, cassava plants are the only plants that stay alive, and many people are forced to subsist on cassava. Unfortunately, eating too much cassava can cause cassava cyanide poisoning, causing a neurodegenerative disorder called konzo. Konzo means tied legs in the Congolese language of the Yaka people, and it causes neurodegeneration of the legs and eventually full paralysis and paraplegia. What's interesting about konzo, though, and those cyanide compounds, is that scientists perfectly understand how they're produced. A team at UC Berkeley is currently trying to use CRISPR-Cas9 to knock out just 2 genes in the genome of cassava that would eliminate the production of cyanide and create a safe cassava variety for the developing world. Unfortunately, the adoption of safe cassava crops in Africa might be a long time coming, as many African governments feel that genetic modification and gene editing is unsafe. Frequently, they're pressured by outside activists from Western countries that try and tell them this, despite the fact that most scientists from those countries know that genetic modification is safe, especially in the case of cassava. It's examples like this why it's absolutely critical that we signal the usefulness and safety of genetic modification for the people that need it the most. So hopefully you see how CRISPR-Cas9 can be a really valuable tool for improving orphan crops like cassava. But I also want to point out that it could be a very valuable tool for improving crops that we grow here in the United States as well. Let's consider just wheat and gluten. Now, looking at a wheat seed here, which is primarily what we make bread out of, we can see that it's really significantly composed of this big proteinaceous body called gluten. Now, gluten is actually made up of 2 proteins, called gliadin and glutenin, and many people find themselves being sensitive to one or both of these proteins. For the majority of people that have celiac disease or non-celiac gluten sensitivity, they're really sensitive to this gliadin protein, which is able to pass through their intestinal lining, triggering an autoimmune response that then degrades their intestinal lining over time. This can be absolutely devastating for people, and really, it's been a focus of conventional crop breeding for decades to try and remove gliadin from the genomes of wheat to prevent this reaction from happening. Unfortunately, conventional breeding has been really unsuccessful at trying to get the gene for gliadin out of the genome of wheat. And the reason for this is because any one variety of wheat can have 45 to 100 different copies of that gliadin gene, meaning that it would take hundreds or thousands of years to breed out all of those genes that were responsible for producing gliadin. Fortunately for humans, Castline provides a really effective means for eliminating gliadin from the genome. One study that was published 2 years ago targeted a Cas9 molecule to all 45 different versions of gliadin inside of the genome for wheat. This was actually pretty easy, because all gliadin genes have a sequence in common that can be targeted with Cas9. What they found is that they were able to actually mutate or entirely delete most versions of gliadin from that genome, and as a result, produced a wheat variety that produced almost no gliadin. What's perhaps most fascinating about this study is that that CRISPR mutant, which had lost most of its functional gliadin genes, looked just the same as conventional wheat. It grew just the same amount of seed, and the seed looked just about the same. But really critically, those CRISPR mutants showed an 85% decrease in predicted immunogenicity. They retained all their bread-making qualities and most of their nutritional profile, but they showed a significantly reduced immune response in celiac and gluten-sensitive patients. Now, it's important to point out that making a gliadin-free, safer version of wheat is totally possible through conventional breeding, but it would take hundreds of thousands of years, and the result would be just the same as if we had used CRISPR-Cas9 and accomplished it in a few months. I hope that this is a compelling argument for how CRISPR-Cas9 and gene editing can be a really useful tool in improving our crops to make them more nutritious and more delicious. Now I'd like to talk about genetic modification with regards to farms and farmers. Now, I do a lot of agricultural outreach here in the city of Berkeley and in the greater Bay Area, and the critique of genetic modification that I hear the most is that it takes power away from farmers and puts it in the hands of multinational seed corporations. Now, I'm not going to have time to talk about that in full today, And I do believe that companies like Monsanto and Syngenta do deserve to be held accountable for the mistakes that they've made in the past. But today, I want to talk about a new vision. I want to talk about how GE and GM can be used to empower farmers, transform agricultural production systems, and protect our society's most vulnerable people. Now, one of the main ways that scientists like me are using gene editing and genetic modification to help farmers is by breeding new varieties of plants that are resistant to fungal, bacterial, and insect pests. Let's consider just one of those pests, shown here, the fungus Fusarium head blight. Now, Fusarium head blight takes out about 30 million metric tons of wheat every single year, and accounts for about $5.6 billion of lost revenue, mostly for American and Canadian farmers. Now, Fusarium head blight is pretty unsavory for two reasons. One, because it can soil crops, but also two, because it produces a really nasty chemical toxin called vomitotoxin. I'll let you kind of use your imagination to figure out what the effects of vomitotoxin are, but suffice to say, it's a really devastating toxin produced by Fusarium head blight that is pretty nasty for humans and livestock. What's interesting about Fusarium, though, is that it's a problem much bigger than wheat, and in fact most species of grasses that we know of are in one way or another susceptible to Fusarium. That is, with one notable exception, the genus Thinopyrum, or wheatgrass, which is a relatively undomesticated relative to wild wheat. If you look at Thinopyrum side by side with wheat, they actually have a ton in common. Their leaves look really similar, their grain looks very similar, they flower in similar times and in similar ways, and actually they're so genetically similar that they can actually breed with each other. They can form a viable hybrid. This would be kind of similar if you took a Labrador and a Poodle and bred them by each other, that you could actually have a viable offspring born of that. But there's one really notable difference between Thinopyrum and wheat, and that's that Thinopyrum has a gene called FHB7. FHB7 is a protein that's able to efficiently detoxify vomitotoxin, making Thinopyrum effectively immune to Fusarium head blight. Now, FHB7 is a genetic oddity. It's the only gene that we know of in plants that's able to detoxify vomitotoxin, which made scientists ask the question, how come Thinopyrum is the only species that has this gene? Where did it come from? They found the answer, and it came from the roots. So, plants, just like you or I, maintain a really complex ecology of microorganisms that live inside of them. We have bacteria and fungi and all sorts of weird stuff that live inside of our gut that help us digest our food and engage with things and bolster our immune system. And very similarly, plants have a very rich ecology of microorganisms that live in their guts, their roots. And Thinopyrum is really no exception. In fact, it maintains a whole network of friendly fungal interactions with fungi that live in and around its root cells. In a stunning display of molecular forensics, one team of scientists was able to show that FHB7 gene, which detoxifies vomitotoxin, 5 million years ago was passed from one of those fungi into the genome of Thinopyrum. This very specific evolutionary event is the reason that all living species of Thinopyrum have resistance to Fusarium head blight today. But what if we could take that gene and passage it into another organism, into the wheat that we lose so much of to Fusarium head blight? Our scientists that did this molecular forensics thought to do just this. They passed that FHB7 into wheat, and what they found was holistic and broad-spectrum resistance to Fusarium head blight. This story is amazing because it shows how a gene was passed from fungus to grass, and in each organism was able to confer a very specific tolerance to vomitotoxin. Now, what I love about the FHB7 story is that it really shows that the transmission of genes from one organism to the other is a natural process. Wheat, just like Thinopyrum, has all sorts of fungi that live in its roots, and it's very conceivable that the FHB7 from those fungi could be passed into wheat. But were we to just sit around and wait for that to happen, we'd probably wait, several hundreds of thousands of years. Whereas if we use Castline, we can accomplish it in a single generation. Not only will genetic modification and gene editing be really critical for feeding generations to come, the things that we want to do with gene editing really aren't that dissimilar from things that happen in the natural world. Now, one of the main questions that I get asked doing agricultural outreach, time and time again, is why are farmers so beholden to seed corporations? Don't plants produce seed such that farmers wouldn't have to buy seed every single year? Now, the answer to that question is complex and multifaceted, but a lot of it boils down to a genetic phenomenon called hybrid vigor. Hybrid vigor works a little bit like this. Let's say that you have two elite cultivars of rice from different parts on the earth. Let's say that one is from Africa and one is from Asia. Now, usually elite cultivars have been inbred for many, many generations, and while they have really nice agronomic traits as a result of this, they've really lost a lot of genetic diversity. But now let's say that you decide to breed those two varieties by one another. And what you find is that the offspring or progeny of those two is actually much higher yielding, much less susceptible to pests, and much more tolerant of drought and other stresses. This is due to a phenomenon called hybrid vigor or heterosis. But there's an unfortunate part of hybrid vigor that when you breed into the next generation, much of those traits are lost, and the offspring of hybrids look very much like the parents that bore the hybrid in the first place. So the reason that so many farmers have to buy seed every single year is that frequently they're buying hybrid seed from seed companies. Plants that will retain the traits of a hybrid plant for a single generation and then become pretty useless by modern day standards. Now, capturing hybrid vigor, that is, producing a plant variety that is able to stay a hybrid for multiple generations, has been an ambition of biologists and genetic engineers for decades, but no one was successful until the use of Castline last year in 2019. A team at UC Davis used Castline to systematically modify a few key developmental genes responsible for rice reproduction. What they had produced was a variety of rice that could produce an offspring without having to be pollinated. It could effectively clone itself through successive generations. What this would mean for the rice industry is that you could buy a hybrid crop and it would clone itself into successive generations and remain a hybrid, still hanging on to those nice hybrid vigor traits. The discovery of plants that clone themselves and the CRISPR-Cas mediated gene editing techniques to do it have blown the doors off of agriculture. Self-cloning plants have long been considered the holy grail of agriculture, and these techniques will be groundbreaking if we can transfer them to other staple crops, like wheat or corn. Stable hybrid lines will be really great for scientists like me, because it'll really accelerate our breeding efforts. Being able to work in a stable hybrid lines, that means that we can really work in a relevant genetic context when we're trying to improve traits in plants. But most importantly, stable hybrid lines will be great for farmers. While self-cloning rice really isn't on the market yet, once it will be, it'll mean that more people can buy elite varieties of rice because it won't be so expensive to have to buy seed for multiple years. This reduction in cost will be especially important for farmers that wouldn't otherwise be able to afford hybrid lines, which will be a really important thing in the developing world as the agronomic demand increases. Now, improved pest resistance and self-cloning plants, I think, are two great examples that illustrate how scientists are trying to develop new ways to empower farmers and to make farming a more inexpensive, practical, and efficient pursuit. Perhaps most importantly, though, gene editing will facilitate the introduction of low-input agriculture. If we can make plants that are drought-tolerant and pest-tolerant and better at nutrient allocation with gene editing, that means that farmers have to use less water, fertilizer, and pesticide, fundamentally putting fewer chemicals into the environment. Now, in this way, gene editing might be our most important asset when it comes to curbing emissions in the agricultural sector. So now that we've talked about some very conventional topics in agriculture, food safety, nutrition, farming practices, I wanted to take a minute to just kind of speculate about the shape of plants to come, how we might be using engineered plants in the future to meet the new problems that society is facing. Now, one can only imagine how humans first felt when they ventured deep into the Brazilian rainforest and discovered glowing mushrooms. The species that you see before you right now is Neonathapus nambi, which is a species endemic to Central and South America. Now, mushrooms like Neonathapus nambi have long fascinated biologists, and right now we currently understand that they glow at night so as to attract bugs that will spread their spores around the forest floor. But it took biologists a long time to figure out the actual genetic mechanism of their autofluorescence. Amazingly, fungal autofluorescence is encoded by just 4 genes, and after several decades of research, scientists were able to identify and get the DNA sequence of all of those 4 genes. In a groundbreaking study that was just published this past year, a team of Russian scientists transferred 4 of those genes and put it into a tobacco plant, and the results were extraordinary. Tobacco plants that fully glow in the dark without the addition of excess chemical energy. So these results speak for themselves, and personally I cannot wait until I have a plant nightlight in my home, but I bring up these glowing plants to make a bigger point. I don't think that these are the most interesting manifestation of genetic modification in plants, and I think that gene editing will do a lot cooler things in our lifetime. But these propose a new paradigm, in which plants are able to be used for tools to solve big human problems. For example, What if you could get a glowing plant that only glowed when exposed to an environmental pollutant? What if you could get a plant that could clean up an oil spill or remove pollutants from a soil? What if you could generate new fuels efficiently from sunlight? What if you could use the roots of a plant to desalinate water? And what if you could mass produce medicine like insulin or a COVID vaccine in the leaf of a plant? Fundamentally, nature has made millions of interesting chemistries and behaviors that are useful to us humans that we could put to use for us in the leaf of a plant. And finally, what if plants could help us solve the biggest problem our society will face in the coming generations? Now, not only is this a huge problem, it's very near and dear to my heart because this is what my research deals in. If you look at this graph, this charts atmospheric CO2 levels over the past 100,000 years on the Earth. What we can see is that as the result of industrialization and the burning of fossil fuels, CO2 levels in our atmosphere have precipitously increased in the past 200 years. Now, what this will mean for our environment is immense habitat destruction, deforestation, and really rising temperatures that will be bad for humanity. But what if we could use plants to help solve that problem? Now, plants, of course, take CO2 out of the air through photosynthesis and convert it into living matter. Pretty much everything that you see around you, whether it's your skin, the leaves on a tree, or fungus in the soil, is made from CO2 that at one point was pulled out of the air by a plant. But in the photosynthesis department, though plants do the most photosynthesis, they're not exactly the most efficient. Consider algae and photosynthetic bacteria, or cyanobacteria. These are the close relatives and even distant ancestors of most plants that we see on land today. Interestingly, because these guys have been doing photosynthesis for so long, they've grown incredibly efficient at it, and actually they're much more efficient at taking carbon dioxide out of the air than most plants. A lot of this is owed to biochemical and biophysical mechanisms called carbon concentrating mechanisms. Here we see a bacterial carbon concentrating mechanism, where the cell essentially packages all of its carbon fixation machinery into a big proteinaceous icosahedral shell, shown on the right, and then surrounds it by a bunch of pumps that pump carbon dioxide towards that shell so it can be more efficiently captured and converted into biomass. As a result of carbon concentrating mechanisms, bacteria and algae are actually much more efficient at photosynthesis, specifically carbon capture, than plants. So this makes scientists like me wonder, what if we could put those mechanisms inside of the leaf of a plant? What if a plant could be taking out more CO2 from the air and converting it into more useful things, like medicine or food? Now, solutions like this might only play a small role into how our society responds to climate change, but even marginally improving carbon fixation in agriculture could have huge benefits for humanity. And I really hope that this illustrates to you that by drawing examples from other parts of nature, we can really improve plants for human good. So today, I hope that I was able to convince you that agricultural crop improvement is still an active process, and that the tools that we have today aren't unnatural or unsafe, but instead just accelerated means to improve the sustainability, nutrition, flavor, safety, and overall agronomic usefulness of the crops that we grow for food. I also hope that I was able to convince you that scientists like me are absolutely concerned with the interests of the people that grow our food and the most vulnerable people in our society that have to live off of subsistence farming. Finally, I hope that I was able to impart and to inspire your imagination about how engineering plants is going to offer really exciting, unique, and scalable solutions to some of the toughest challenges our society will face in the coming decades. Now, as the sun sets on this presentation, and the sun literally sets all around me, I wanted to make just one last point, and it starts with this image. This is a photograph taken in the 1890s in the United States, and what it shows is a team of dozens of horses pulling a piece of agricultural equipment. Now, nowadays, horse-based agriculture seems entirely impractical. Horses have long since been replaced by tractors, but in the period following World War I, Americans had a very different perspective. As mechanization slowly was replacing the jobs of labor animals, heated political debates were ensuing, arguing that tractors were unnatural and inhumane. One lobby group, called the Horse Association of America, even sprung up and spent the better part of the 1920s and the 1930s spreading fear and propaganda and going around the entire United States talking about the march of machinery and the scourge that tractors brought to agricultural life. These tensions around agricultural mechanization really came to a head at the 1921 National Implement and Vehicle Association show in Fargo, North Dakota. At the show, a tractor was pitted against a team of 60 horses to see who could faster prepare a 10-acre seedbed in sweltering 100-degree June heat. Perhaps not so surprising to us today, the tractor smoked the horses, and not only did it win by a landslide, actually 5 out of the 60 horses died of heat stroke. I think stories like this are a really good allegory for genetically modified crops, because in most cases, genetically modified crops are safer, more efficient, better for farmers, and significantly more humane than those bred with conventional agriculture. But just like people were stubborn to horses being transitioned out of agriculture, so are people stubborn about the introduction of genetically modified crops into our food supply. And I think that this brings up an important point, and that's that change in agriculture is really hard. Riotous public debate has coincided with almost every transition in agricultural history, whether it be the transition from oxen to horses or the transition from manure to fertilizer, which is why it's important that we communicate to everyone that this technology is not only critical for meeting the needs of our growing population, but it's safe. But I don't ask that you just take my word for it. I'm just one scientist, and science is a community effort. Instead, defer to almost every major scientific organization on the Earth, like the World Health Organization or the National Academy of Sciences, who have all concluded that incontrovertibly genetic modification is safe for consumption. It's these same organizations that you trust with regards to climate change and vaccine safety. Quite simply, the opposition to GMO stands with blatant disregard to the evidence, and a growing scientific consensus suggests that GM will be one of the best tools that we have in meeting the needs of our growing planet. Now, this doesn't mean that reasonable people can't disagree about specific agricultural practices or how we might use gene editing. But our global situation necessitates that we reshape the argument about GMO, not to be about if we should use it, but about how. So I'll leave you with just one last point, and that's that fundamentally, you as an American consumer will never face the need to eat GMO. But that's an immense point of privilege. Mind you, I dream of a world where you would be so attracted to eating GMO, whether it's more nutritious or more delicious, that you would want to do that, but American agricultural output will be never as affected as the rest of the world by climate change. But the world is looking at us as an example when it comes to agricultural development. When we oppose GMO, whether it's in our legislature or on our food labels, we tell people that need this technology that it isn't safe, which science tells us is just not true. To oppose GMO is to let the vague and largely unfounded fears of the few stifle the prosperity and the livelihood of the many. Now, I hope today I've excited your imagination about the coming good in our world, given you tools to understand this coming age, and imbued your trust in this brave world to come and in the scientists that are working to make it happen. Thank you so much for your attention. This has been an immense blast, and I am really excited to follow up with you all in the chat bar. Thank you to Wonderfest again. Please hit me up. Would love to talk to you after the fact if you want to email me or tweet at me or check out the other stuff that I'm doing. Thank you so much for your attention. Goodbye.
Everything is new. Why not just ask why? Why can't I see at night what is said anyway? Why must people fight? Maybe it's just our way. No one really knows why gravity exists. Naming something doesn't mean that we get it. So I'll live each day with